Hello and welcome, everybody. The presentation today is titled The Enemy Within, How Hospital Mattresses Act as a Hidden Fifth Column Undermining Patient Safety in Healthcare Facilities. So you may have heard of the concept of the fifth column. The fifth column comes from the Spanish War, and also from World War II, where we talked about internal saboteurs of threatening our columns of liberty, unity, justice, and equality here in the United States. Well, now we talk about the fifth column in healthcare. And we have pillars, again, in healthcare, the quadruple aim: better healthcare outcomes, improved patient experience, improved staff experience, and lower cost. But we also have that fifth column attacking these other four columns. That's the hospital mattress, the number one touch point in hospitals. So an average acute care bed in a hospital will support about 80 patients a year, okay? And then those, I call them the Trojan horses, move from room to room about 3 times a month, okay? So they're moving around the hospital and carrying the bacteria with them. And there was a study done by Medline with over 5,000 mattresses, and they found 59% of them had issues. And we'll talk about that some more. And in a previous study I did, we found that 52% of infection practitioners said that they had had leaking mattresses and exposing patients to patient bodily fluids. So the CDC kind of got involved in this. You know, they've emphasized forever, and as has all of us, you know, the importance of hand washing. But in 2020, they were like, you know, hand washing is not the only thing we need to worry about. We've got these contaminated surfaces that are being the source to contaminate the hands. And the problem is we all fight with trying to get improved hand washing in our facilities. But the issue is that's trying to change human behavior. We're going to introduce today to you an engineered, easily done solution that can save patients' lives by decreasing infections.
So let's just take a minute to own how complicated it can be to do the right thing for patients when it comes to reprocessing mattresses. This slide shows that a variety of stakeholders involved in the cleaning and disinfection process. The EPA regulates disinfectants, but the FDA regulates medical devices. The CDC and FDA have standards for disinfection, but they leave it up to the bed and mattress manufacturer to establish the instructions for use, misuse, and choose and validate the choice of disinfectant. These manufacturers determine, too, what is meant by disinfect to the worst case scenario for that medical device. In a hospital world, does that mean an isolation patient? Or is the worst case the patient that's normally on the bed? The hospital is required to follow the instructions for use, although the hospital can take additive steps to the instructions for use. As an example, many hospitals began using fluorocidal disinfectants daily to protect patients, or to protect the patients from asymptomatic C. diff patients. However, those additive steps may not have been considered by the device manufacturer. As an example, a major manufacturer just recently recalled 58,000 mattresses due to the extensive use of bleach, and have since changed their instructions for use to add a rinsing step before and after bleaching steps. The point is, while the manufacturer understands the material it's using, it doesn't always coexist with the additional steps that a hospital may choose to go beyond what the manufacturers describe. In this webinar, we tried to capture clearly the objectives we're all trying to achieve in patient safety and the vulnerabilities inherent in this siloed system. But if it feels complex, don't worry, it is. Next slide. The learning objectives today are to to focus on healthcare mattresses, beds are an issue as well. The objectives are to dimensionalize the size and scope of the mattress problem, to share data on its role in patient safety, to explore why the mattresses aren't getting cleaned, and to understand what compliance looks like for cleaner mattresses and therefore improve patient safety. My name is Bruce Rippey. I founded Trinity Guardian some 14 years ago and I'm the CEO of the company. Today, I'll be joined by three experts: Dr. Hooker, who started this webinar. Dr. Hooker is an epidemiologist, an ER physician, professor at Xavier University's Health Services Administration program, and is a medical director for Trinity Guardian. Dr. Artis Hoban is a recently retired University of Kentucky professor in internal medicine and infectious disease, former member of the Kentucky Health Cabinet in infectious disease, as well as practitioner. He is also the past president of the AMA and is a Trinity Guardian board member. Barbara Strain is a clinical microbiologist with her own consulting firm. She retired from the University of Virginia Healthcare as Director of Value Management and is an advisory board member for Health Services Institute.
So let's talk about the size and scope of the problem. The down and dirty on dirty beds, okay? And beds are really dirty. So, you know, a picture's worth 1,000 words. These are actual healthcare mattresses in hospitals. You can see the soiling on the far left picture. It's embedded in the fabric. I bet you a bunch of you have seen that on your own beds. The middle one is, if you open the bed, you can get to see all the blood, gore, and more inside. That red stuff, I think we all know what that is, okay? And so that can go into the mattress and come back out of the mattress onto the patient. And then the one all the way to the right shows the cover actually delaminating is what we call it, but I'll call it disintegrating. From the disinfectants, these heavy-duty disinfectants we've been using, and also it's destroyed the fire barrier that every mattress is required to have. So no longer is the mattress clean, safe, it's also a fire risk. And here's some more lovely pictures. The pictures on the left shows the, again, delamination. It shows the fire barrier failure, and then you can see those lovely staining of the fire barrier. That was the patient on this bed right before these pictures were taken was C. diff. Delicious. We also have the upper right, we have holes in the foam underneath the cover, and the fluid is being absorbed into the foam. And then in the bottom picture, we have air loss, low air loss mattress opened after a C. diff patient discharge. And again, you can guess what the brown color is, okay? I don't really want to. Next slide, please. And it's not just one dirty hospital that we went and found, okay? This is a problem across the United States. Medline did a study of 85 facilities with over 5,000 patients, and 59% of the beds had problems: holes, tears, stains, internal damage, thinning areas, torn zippers. Huge problem. This is everybody's problem. So we did a study of a major medical center. We examined their beds, all their beds, and we found notably 72% had failed. Most of those were tears and problems with the surface cover, but almost 30% had fully failed with stuff inside of the mattress. Well, guess what? 75% of those mattresses were less than 4 years old. So if you think, oh, I replaced my mattresses a few years back, I don't have a problem, you are not right. It is a huge problem. So there has been a lot brought back to validating this concern. We started this really with the FDA about 10 years ago. They issued a warning, said mattresses are failing. We're exposing patients to body fluids. Then ECRI said in 2019 that this was one of the top health technology hazards. Washington State nurses last year at St. Joe's were concerned that the hospital was continuing to use mattresses for patient care that had body fluids inside of them. And then most recently, the EMRI study, they showed that Clostridium difficile is really related to the bed. And so this was a problem. But what was really concerning about this was that up to 90 days after the index patient, you could get an C. diff infection from that patient. So the C. diff spores are just hanging around waiting to be a safety hazard for a patient. In that same 2019 study I mentioned previously, 52% had a mattress failure, and 2/3 of those people had multiple mattress failures. So, a lot of people use quaternary ammonia to clean, and we did a study back more than 10 years ago where we showed that although it did decrease the bacterial counts, it only did it by about 90%, or that infamous log 1 reduction. Well, what does that mean? Well, it means that there's tons of bacteria all over that mattress. We just took a teeny little spot and looked at it, okay? But now multiply that by the entire mattress, and we've got thousands, tens of thousands of bacteria exposing those patients. And those bacteria continue to grow. We didn't kill them. So now there's a study that showed after about 6.5 hours, there would be recolonization up to 42% of the original colonization on that mattress. So if you don't get it all, you got a problem.
Thank you, Dr. Hooker. Now we're going to talk a little bit about the regulatory guidelines, and we're going to specifically talk about the 2015 reprocessing guidelines that FDA put out. This table sort of helps you to see where the problem gets started. It's a variety of different steps. So when we talk about soil in the first column, that means what comes into contact with these mattresses over the time that a patient is in that episode of care. That then defines what level of disinfection might be required, as well as the objective of that particular disinfection method, and then the required impact. So there's 3 different levels of disinfection. And when the patient is in contact with the mattress, that may be non-intact skin from the patient, which may be a big gaping wound that may be infected, and that requires high-level disinfection. If blood or body fluids, as Dr. Hooker explained in some of those visuals, then that becomes an intermediate level disinfection. And always normal patient flora from their skin sloughing and a variety of other things come from the patient, and it's considered low-level disinfection. The objectives of these 3, the high level must be free of all microorganisms with small spores remaining. Then the medium, intermediate level is killing viruses and mycobacteria and vegetative bacteria, as well as the normal flora is free of vegetative. So they have a lot of things in common, but mostly in common is the impact must be at that 99.9999% level, or else you get a variety of issues that we're going to discuss next. The blood culture plate, the agar plate on the right, is bacteria that grew from a clean mattress, meaning it came from a room that was terminally cleaned in the way that is prescribed from the manufacturer instructions for use. On the left, we talk about that log reduction that Dr. Hooker mentioned. So at 90%, which you might think is a great kill rate, if you started with 1 million microbes, you'd have 100,000 left, and so on and so forth down through the logs. So even if you're at 99.9999%, you still have potential of one organism left. I did a rough count by finger, and there's probably close to 200 on this plate left. So as we look at that, we know that other bacteria that was isolated in a study that you can read that Dr. Hooker published, these were a variety of other microorganisms that were left after that terminal cleaning. And you'll recognize some of these bolded that you hear occasionally of MRSA, BREs, Acinetobacter, Klebsiella, Pseudomonas. These are all potential causes of hospital-acquired infections that we know of. So it's important that those instructions and that terminal cleaning is done. Now, if you think about this, that terminal cleaning may occur after 4 days of patient and they're discharged, 2 weeks, a month, depending on how long they're there. So there is a long time between the fact that you can terminally clean. So at the minimum, every time a patient is discharged or transferred to another unit or a nursing home, that mattress must be terminally cleaned. Now we'll go in a little deeper into microbiology, because when Dr. Hooker mentioned colonization, one of the things that's crucial is the role biofilm plays in spreading healthcare acquired infections. The first step is if you see a few bacteria, the vegetative microbes, they may not attach, but when they go into the second phase, within seconds and minutes, these first colonizers become irreversibly attached to the surface. Then in step 3, they grow, they have cell division, and this can take a matter of just hours. It could take a longer period of time, like days. But then they start to produce this EPS, or this extracellular polysaccharide matrix. This is what's critical because it actually surrounds the bacteria and protects them from the natural body defenses, as well as any antimicrobials that may be injected into the body and others, so that they stay protected for hours or days. And then they can reattach in other places. So you have wet biofilms, which we've known for a long period of time with drains and sinks, but any surface that stays wet, and each type of surface becomes its own challenge. In the more recent years, we've also discovered that there are dry surface biofilms, and the frequency of doing wet cycles and dry cycles can affect the formation of biofilm. So this means dwell times, length of doing the cycles, making sure that they're dry. And medical devices, so in this case, we've been talking about mattresses, also are critical in forming biofilm. So the design of the device, the manufacturer's instructions for use, the damage that it may cause, and useful life. So in these reprocessing guidelines, it specifically lays out how all of these need to be validated through specific testing by the manufacturer for their device, and included those instructions for use in order to have good cleaning.
Thank you, Barb. So there are just an absolute ton of studies that are showing that hospital beds aren't clean when they are, quote, terminally cleaned. They are still teeming with bacteria, and this has just been proven over and over. So why do we care? Because everybody on this webinar cares about patient safety, and this is a huge patient safety issue.
So thank you, Eddie, and you are very correct on this issue. This is a very much underappreciated and unfortunately frequently unrecognized patient safety issue. We know that the patients are exposed. They are a number one touch point in that bed, and number two for healthcare bedside caregivers, and they creates multiple opportunities for infection. So we know that these damaged mattresses are common and lead to infection. And this particular slide just demonstrates for you all the number of studies investigating this particular issue. But let's move forward and look at some more recent data in this particular area. In 2018, there was a study published in Infection Control and Hospital Epidemiology by Cohen et al., and it revealed a patient was 5.83 times more likely to get a hospital-acquired infection if the previous person in that bed had an HAI. And if the patient was 2 to 3 times more likely to acquire a C. difficile infection if the previous bed occupant also had C. diff. Those are really frightening numbers. And then most recently, the ESCAPE study, again showing the multiple organisms, all of which can become pathogens, can be nosocomial and associated with high levels of resistance and increased morbidity and mortality. So let's look at this next slide. It provides some recent information and some studies related to mattresses and their problems associated with Enterobacter cloacae and Klebsiella. And I would point out to you that in the first one, 4 deaths, 18 infected or colonized, that colonization rate, again, infected or colonized, is very important because as all of us recognize, colonization with these high pathogenic pathogens can lead to serious infections and death. Again, as reported earlier, all mattresses were less than 18 months old. They were considered new mattresses, and you can see there the data. And then in the third column with Klebsiella, 19 people became ill from a single bed unit. I find this absolutely terrifying. 19 people exposed and became quite ill during their time on that particular bed or their mattress. Next slide. So this particular slide reports on the Emory bed study, which is published in 2023. And again, some very impressive data. This demonstrated a 50% increase in C. diff if the previous patient had C. diff. You saw that in an earlier slide. And despite the cleaning efforts, those beds were still dirty after 3 months period of time. This coming from a highly reputable institution, very perceptive in what needs to be done. This is a real world study, and it shows what can actually happen. We also note that UV light and bleach did not break that cycle. And again, there were multiple interactive factors. You've seen previous slides that Barbara just showed about biofilms and such, lends itself to a variety of issues that have to be addressed when we're doing concepts around cleaning and terminal cleaning. This final slide here on shows a VA hospital safety alert. You can read the details. This was issued in April of this year, 2024. And again, it talks about significant risk that can lead to hospital-associated infections, patient tissue degradation, serious illness, and then ultimately potential patient death because of the egress and ingress of bodily fluids. And then you see in this particular summary slide, this risk has been identified significantly by multiple organizations. The VA, as you just saw, the FDA, ECRI, the American Medical Association, the Centers for Disease Control, and CMS as well.
So why aren't the beds clean?
Thank you, Artis. I appreciate it. So why in the heck is this going on? Because everybody cares. Everybody on this webinar wants the right thing for the patients. So let's examine some of this. Next slide, please. So there's been an evolution. There's been an evolution of the mattress. We all remember the 1965 Chevy where we had a vinyl seat and we stuck to it. Okay. Well, the problem was that we had vinyl making for all the mattresses, and there was a link believed to be between that and bed sores and patient skin breakdown. So we created these new types of mattresses that can breathe, and they're called microclimate. And so instead of that nice thick non-porous vinyl, we now have thin 1/40th of an inch thick is the new mattress covers. They are so thin it is not believable. And although they're intended to breathe, that makes them porous, so we can't clean them like we clean the vinyl. And the expected life has dropped from 10 years to 1 to 2 years for these new mattress covers. So that's the first thing that's changed. The second thing that's changed is the bugs. The bugs are not very nice now, the MDROs, okay? They are not killed with a lot of our older disinfectants. So we've had to start using bleach and start using all sorts of very strong disinfectants in order to get these mattresses clean. The problem is not one of those is actually approved for use on a porous, soft surface like every mattress is now. And so there is a misperception about cleaning, okay? And so I want to kind of do a little survey here, if you help me. Next slide, please. So in your facility, are you following the manufacturer's instructions for use? If you don't know, say you don't know. But if you absolutely know, please mark on your screen whether you're following it, you know what they are and you're following them. No, you don't follow the instructions for use, or you're just not sure. And I will give you about 30 seconds to answer that. We'd appreciate it. So about 18% are following the MIFUs, about 16% aren't, and 66% weren't sure, okay? And then you've already jumped and started answering the second question, which will give you plenty of time. But based on what's being responded to now is most of the people are using a one-step process where you are wiping with one wipe, cleaner, disinfectant, all done, is what's going on there. And then we asked also about bleach, you know, some of your facilities are using bleach, some are not, and many are not sure. And this is not surprising data, okay? I'm gonna close this thing. This is not surprising data. If we could forward to a couple of slides, Nicholas. Okay, so we did a study back in 2019 where we asked everybody, and 86% at that time were using a one-step process. It sounds like people are continuing that know what they're doing, that know what their facility is doing. But also, I think from what you all just responded, very interesting that many of you don't know what your EVS workers are doing, okay? Are they following the instructions for use? So let's continue on.
Thank you, Dr. Hooker. And now we're going to talk about those, what we call 5 elements of getting a bed clean. The first one is so important because you have to recognize the type of bed, which means that each manufacturer and each type of bed has its own manufacturer's instructions. Now, given any hospital, you might have 2, 3, or more different manufacturers of beds, and you might have different types related with each of those manufacturers. Each of them come with their own IFUs. And some manufacturers do have a 5-step process, and we'll go through that in a little bit. And some have a 6-step process. So to recognize that is so important. Then you need to recognize that soil level on that bed. What has happened when the patient has come in contact with that mattress surface? Is it normal flora, body fluid, wound exudates, or do I just choose the worst case scenario in order to satisfy that chart about 99.9999%? And hospitals sometimes augment this by saying, is it an isolation room for a specific organism like C. diff or a multiply drug-resistant organism, or is it non-isolation? So ECRI also suggests notifying environmental services about what the type of bed and the soil is, but if that's not possible or part of the policy, you must default to that worst case scenario in the circumstance. You then have to choose the appropriate cleaner and disinfectant based on the manufacturer's IFU. Each of those may be different depending on the validation testing I referred to earlier that was done through those reprocessing guidelines that must be followed. So there's a difference. Then it's human elbow grease, as we're calling it. Every person who does this function, the soil must be removed first, and that takes effort to do that. Also, what's the cloth that's being used, and what's the direction that the activity is happening? And then the last 5th element, it's a multi-step process. So you've got to clean, you've got to disinfect, and you've got to allow for the dwell time that's actually on the disinfectant or the cleaner packaging. And this dwell time is really important. So the MIFU 5-step really looks like this. You have a pre-clean in which you remove gross soil, you clean non-visible soil, assuming that it's had this contact. If you've used bleach, then there's a rinse step. Then you apply the disinfectant, and this might take either 2 or 3 rewipes to make sure that the dwell time is accurate. And then there's a rinse step at the end so that damage isn't done to that surface of the mattress.
So we have another polling question for you. Does your facility use bleach as its disinfectant after each patient discharge for mattress reprocessing? Nicholas, if you could call that up, we'll let everybody vote real quick.
Bruce, I think a lot of people already voted. I think they got all of the polls. I don't know how to see the answers if that poll could be flipped up.
So we'll come back to that. Bleach works, but again, not as a one-step wipe and walk process that many would be using today. The FDA reprocessing guidance, as Barbara described, indicates that any medical device surface must be cleaned before the disinfection step. These are very important. Again, you have to pre-clean the gross soil, remove the visible soil, then clean the non-visible soil, and then potentially a rinse before the disinfection step, and then rinse again. Next slide, please. In the real world, these processes don't always work as intended. And when the IFUs are created or when the disinfectant labeling is created. And this is an example of it. The Manion study several years ago reported that after 4 rounds of cleaning with bleach in rooms with the Acinetobacter and MRSA, that only that 1 out of 4 rooms still remain contaminated. It's essential to rinse following the bleaching step. The final step is important because while the hospital disinfectants can be used according to the IFU, they're still intended for hard, non-porous surfaces. And they will eventually cause the fabric to deteriorate, as you see in this SEM image above. In fact, in trying to reconcile mattress reprocessing with IFUs between the device manufacturer and the disinfectant manufacturer, it's important to remember that it's the bed manufacturer's IFU that controls. From a risk management standpoint, there's no consensus document or evidence-based guideline that promotes the use of a hard surface disinfectant on a soft porous mattress. It's a one-step process. In fact, one bed manufacturer has recently updated their IFUs to indicate that only 6 bleachings can be used over the entire of the mattress over the entire life of it. There's literally dozens of chemical formulations of coated fabrics, and they're as different as Coke and lemonade in terms of compatibility to these disinfectants.
Unmute. Thank you, Bruce. So people have tried using hydrogen peroxide vapor. It does get a little bit increased decrease in bacteria, log 2 to log 3, but not log 6. And the problem is it's not been proven to work on porous surfaces. And the other issue is it's not practical. You've got the bed out of service 4 to 6 hours. You have got to tape all the vents shut. And it also can damage the paint on the walls. It can damage the devices in the room. So though it does actually increase kill of bacteria, it's got a lot of issues in the real world. So UV light. UV light, the FDA says it's not intended for soft porous surfaces. The new mattresses are all soft porous surfaces. No bed manufacturer recommends using UV light to disinfect their mattresses. So you're not following the IFUs if you're thinking you're doing it. And the only randomized trial of UV light showed it did not work to decrease C. diff infections. That was the Anderson or Better D study. So is this me? Okay. Okay.
Hey, thanks, Eddie. I think everybody realizes that when we understand the pathogenicity of the many organisms we've been talking about today, we want to be as aggressive as possible in dealing with these on these beds and on these mattresses. On the other side of this, we also know that a one-step wipe and walk simply will not work, and it does not meet the FDA or CDC requirements for disinfection. So in the presence of things like these multiply drug-resistant pathogens, these organisms we've been referring to, in addition to softer porous mattresses and the need for disinfection at terminal cleaning, we've got to be more precise about what we are doing. Yes, the methodology that's been looked at, that you've been exposed to during this presentation is complicated, but in fact, we need to deal with this in a much better way.
So what does a clean bed compliance look like? Basically, it's following the manufacturer's IFUs, and this is supported by various ways. So the regulatory philosophy has changed significantly. So those 2015 reprocessing guidelines we've been talking about eliminated what was an option to follow facility protocol. That might be coming from past practices where you might say, let's use a quats in every situation, except if we have C. diff, let's use bleach. Some sort of generalization, so it sounds like it's easy to follow. But that's been debunked these days. And the CMS and Joint Commission have all circumscribed reprocessing options as follow the manufacturer's IFUs only. So that's why those are really important. The FDA has put out a guideline about keeping patients safe from contaminated mattresses. And there's really 4 steps to this. Is 1, you make an inspection plan. So there's detailed steps about when and who and how. Then you have to actually do the inspection and follow your plan. And then when you find issues, as was discovered in the Medline and other studies, then you must remove and replace any damaged mattresses, mattress covers that you find. And then now that you have refreshed all that, you now need to maintain all of that by following the manufacturer's IFUs. So that's repeatedly the CDC, also in their guidelines for infection control, have also repeated those. The Joint Commission in July 1st of 2024 has re-emphasized that following the manufacturer's instructions for use in bed and mattress reprocessing is a requirement. You must follow it for disinfectant use. Do not use anything off-label to the manufacturer's IFU, and ensure that you have that management plan for your mattresses and you are following it. So meaning you have to do a lot of documentation in there to say that I've done these things. So it's important to understand that each patient care item, regardless what, has its own IFU, cleaning, disinfection, and then the expectation that the organization follows those. So these are very critical steps.
Bruce.
I'm sorry. So the next step from leading manufacturers are to follow, as we've discussed, these pre-cleaning steps of pre-cleaning gross soil, removing non-visible soil, rinsing, disinfect. And that can take 2 to 3 wipes depending on the dwell time. And again, you'll notice there's many other issues concerning this from a compliance standpoint in terms of just the human interaction in the process of doing this. Next slide, please. So this slide shows what the dwell times are and the time it takes to use these individual disinfectants. And you'll note that all of these are going to add significant reprocessing time to the mattress. If you're using a one-step process right now, understanding to be compliant with the MIFUs is probably going to add 8 to 15 minutes to your room turn time. And you have to, again, keep in mind that these beds are traveling on average 3 times a month and accommodating different patients. So these are really a big issue from that standpoint. And you can, again, your concern is the human factors, how much elbow grease is in this. Just an example of the human error in reprocessing. Just this past week, a major hospital revealed that there was an error in scope processing or reprocessing endoscope that may have exposed patients to HIV or hepatitis. The challenge is educating your staff to comply with these complex reprocessing steps in a repeatable manner. And that's a very difficult challenge with the turnover that you have in EDS departments today. Another really important point that most hospitals don't get right is understanding those IFUs require that integrated cover to be replaced at the end of the expected life. And when it starts to fail and moisture builds up inside the mattress, mold can grow. The manufacturer's warranty is then voided if the integrated cover is not changed at the end of expected life, leaving patients vulnerable.
So briefly, in summary, you have to follow the FDA, the CDC, and CMS guidance. The messaging has been consistent on this. you've got to follow the bed and mattress manufacturer's reprocessing MIFUs to be in compliance. And if you fail to follow the MIFUs, your mattresses are going to get degraded. It's going to invalidate your mattress warranty, and it will jeopardize patient safety and potentially cause a patient death. So, what does compliance look like? Let's talk about the Soteria bed barrier. The Soteria bed barrier is the first of its kind launderable and reusable healthcare bed barrier that provides a physical barrier to protect the bed and the mattress from patient soiling, helping to reduce contamination during use. You can see more of the details there. But right now, I want to turn this back over to Bruce and let him talk about this. This is something he's been very passionate about. Bruce?
So we recognize that beds and mattresses have been contributing to hospital-acquired infections and represent a significant patient safety risk. Nobody wants to see their loved one laying on a mattress that is oozing fluids. And our goal in developing this product has been to develop a better way to reprocess a bed. The FDA assigned our bed barrier a unique product code when it was cleared in 2022 due to its innovative nature. And it is the level 4 barrier. Our product simplifies this entire silo of these individual silo issues between the EPA, the FDA, the bed manufacturer, and it helps give a standardized solution. Ours is an engineered solution to minimize the human factor, variability, and reprocessing. You can see on this slide a patient discharge. EVS can simply release the barrier from the mattress and bed deck and fold the barrier into itself and placing it into a biohazard bag. The magic is not only the material used in the bed barrier, but a proprietary validated laundry process that can be accomplished in either the healthcare facilities in-house or outsourced laundry. The laundry process was validated in the performance testing for the 510K and in peer-reviewed randomized clinical study. It's laundered using a 160-degree water temperature and chlorine bleach. The final step is pH to make it pH neutral. It's then light table inspected between each use, and it's guaranteed for 150 uses and is RFID chip for tracking purposes. The laundry process achieves that goal of a 99.99999% level of clean. The FDA's equivalent to high-level disinfection. This Petri dish illustration describes that further. This also includes Pseudomonas, MRSA, C. diff, and Klebsiella examples. Ready?
Thank you, Bruce. So, the issue is, what do we get from this? Well, if you use just standard terminal cleaning, the mattress still has bugs growing on it. We did a study. We showed that it decreased about 90%, but there were lots of bugs still on the mattress. If you put a Soteria bed cover on there, or bed barrier, the mattress did not become contaminated. There was the same amount of soil before and after. So this is amazing. This keeps the mattress from becoming contaminated by that patient that might have C. diff. And what we did in the real world, we were able to decrease C. diff infections in real patients by 50% and 2 LTACs. So very sick patients. So the cover is amazing. It's breathable. It's a 4-way stretch fabric. It's free of forever chemicals. It's made of the same material as the current high-level air loss mattress, low air loss mattresses. And guess what? Importantly, no increase in skin damage. So no increase in skin injuries. Bruce?
Yeah, so recognizing the vulnerability of infants with fragile skin and lungs, GE recently updated its instructions for use for incubators, now requiring high-level disinfection with multiple sterile water rinses. as an option, they offer this recommendation, and that is directly from their instructions for use. It is recommended to use a commercially available microbiological barrier with appropriate decontamination level. The Soteria neonatal mattress barrier is the only commercially available barrier on the market that's been validated for use by GE. We do have a cost calculator. The typical calculation is to look at value as the quality divided by the cost. We do have a cost calculator that can help with this. Suffice it to say that in general, facilities have seen that it costs generally less than $3 per patient day. For perspective, a hospital-acquired C. diff case today is averaging about $34,000. Litigation for healthcare-associated infections is significant in terms of settlements from state court actions, because oftentimes it goes beyond just the infection itself. Is artist related, it can end up in death or other complicating factors. And we can also reduce the mattress failure rate as well as extend the expected life of the mattress. Next slide, please. So consider this product for certain areas of the hospital to get started. Look at isolation areas, look at high-risk patient populations, ICU patients, NICUs where you have highly vulnerable patients. Next slide, please. You can count, what you can count on with our product is a 6-log reduction of bacteria, including spores, a reduction of your room turn time compared to the instructions for use, and to help increase the life of your mattress. I would encourage you, as we wrap up in this webinar today, as you're looking at how to move forward with your mattresses, and that is to look at the process to ensure that you're getting at least an intermediate level of disinfection, that you discuss that with your bed manufacturer on how to achieve that and how to clean and disinfect for C. diff that has been validated by their instructions for use, as well as what is the integrated mattress cover life, and to make sure that you're replacing those covers at the end of that life, because we know that they will leak at that point. ECRI has recommended that if a manufacturer cannot provide that information, consider alternative manufacturers' products that do have complete instructions for use. An IFU that states follow facility protocol or follow a disinfectant manufacturer's labeling instructions has not likely been validated by the manufacturer to meet the FDA requirements. And I would also encourage all the attendees today, make sure you have an internal mattress inspection plan and that it's followed, because it's a key issue to patient safety. And so with that, I'll be glad to answer any questions that you all might have. I do see one question. Do you have different size barriers for peds, NICU? The answer is yes. Are there any other questions? Someone asked if we would provide samples. Yes, if you will reach out to us at info@trinityguardian.com, we would be glad to chat with you about that.
Bruce, while we're waiting on any further questions, I would just encourage, since we had so many people that don't know what their EVS teams are doing, I would recommend highly that you find out, because you might be shocked and unhappy to find out that they're not following the instructions for use from the manufacturer. They're following some sort of facility protocol that they develop, which does not meet the CDC, or CMS or Joint Commission's requirements.
If anyone has questions after this, you may also submit them to info@trinityguardian.com. If you have questions to any of the speakers today, we'll be glad to forward those to them as well, and they can respond to you directly.